Eukaryotic transcription. Here we could see a picture which depicts this transcribed DNA strand, RNA polymerase, inactive DNA strand, then newly formed mRNA. So, eukaryotic transcription. Eukaryotic transcription is the elaborate process that eukaryotic cells use to copy genetic information stored in DNA into units of RNA replica. A eukaryotic cell has a nucleus that separates the process of transcription and translation. Eukaryotic transcription occurs within the nucleus, where DNA is packed into nucleosomes and higher order chromatin structures. The complexity of the eukaryotic genome requires a great variety and complexity of gene expression control. Eukaryotic transcription proceeds in three sequential stages: initiation, elongation, and termination. The transcriptional machinery that catalyzes this complex reaction has at its core three multi-subunit RNA polymerase. Eukaryotes have three nuclear RNA polymerases, each with distinct roles and properties. Like RNA polymerase 1, pole 1 or pole A, RNA polymerase 1. Location: nucleolus, product larger ribosomal RNA or rRNA, 28S, 18S, 5.8S. RNA polymerase 2, pole 2 or pole B, nucleus, location. Then product messenger RNA, most small nuclear RNAs or snRNAs, small interfering RNAs or siRNAs, and microRNA, miRNA. Then RNA polymerase 3, pole 3, nucleus and possibly the nucleolus, nucleoplasm interface. Transfer RNA, other small RNAs, including the small 5S ribosomal RNA, snRNA, signal recognition particle RNA or SPR RNA, and other stable short RNAs. RNA polymerase 1, pole 1, catalyzes transcription of all rRNA genes except 5S RNA. These rRNA genes are organized into a single transcriptional unit and are transcribed into a continuous transcript. This precursor is then processed into three rRNAs, 18S, 5.8S, and 28.0S. The transcription of rRNA gene takes place in a specialized structure of the nucleus called the nucleolus, where the transcribed rRNAs are combined with proteins to form ribosomes. Promoter structure for RNA polymerase 1. Genes for ribosomal RNAs are exclusively transcribed by RNA polymerase 1. In eukaryotic system, most active and highly productive genes which are transcribed most of the time are ribosomal RNA genes. More than 90% of the total RNA found in any eukaryotic cell is rRNA. It is synthesized as triggered when cells are activated for cell proliferation. In such situations, tremendous increase of rRNA takes place. rRNA synthesis during oogenesis is a par excellent example. UCE, core element transcribed region. UCE and UCE region, then core element and transcribed region. Then initiation. It has what is termed as core promoter region between 10 and 45 and an upstream control elements UCE. Upstream control elements, we just mentioned it earlier. UCE and upstream control elements. Upstream control elements. It is the region to which upstream element binding factors bind. The core region attracts selectivity factor SL1, 3 TAFS, TBP associated factors, and TBP tata binding factors. TA box. Positioning of the TBP is assisted and determined by the SL1, and then TAF bring TBP. It is now known that two histone-like proteins are also associated with this complex. This assembly ultimately brings RNA polymerase 1 to the site, but the activation depends on upstream control element binding factors UBF1. They bind not only to the core but also to UCE. So UBF1 binding results in protein-protein interaction in such a way 2 units of UBF join with one another with a DNA loop and activate the RNA pol 1 complex. UCE and core element pre-rRNA gene. UBF regions, TF1, pol 1, etc. Next, elongation process. As pol 1 escapes and clears the promoter, UBF and SL1 remain promoter bound. UBF and SL1 remain promoter bound, ready to recruit another pol 1. Indeed, each active rDNA gene can be transcribed multiple times simultaneously. Pol 1 does seem to transcribe through nucleosomes, either bypassing or disrupting them, perhaps assisted by chromatin remodeling activities. In addition, UBF might also act as positive feedback, enhancing pol 1 elongation through an anti-repressor function. An addition factor, TIF1C, can also stimulate the overall rate of transcription and suppress passing of pol 1. As pol 1 proceeds along the rDNA, supercoils form both ahead and behind the complex. These are unwound by topoisomerase 1 or 2 at regular intervals, similar to what is seen in pol 2 mediated transcription. Elongation is likely to be interrupted at sites of DNA damage. Transcription coupled repair occurs similarly to pol 1 to transcribed genes and require the presence of several DNA repair proteins such as TF2H, CSB, and XPG. So RNA polymerase, promoter, sigma factor, then attachment of RNA polymerase, then terminator, unzipping of DNA, movement of RNA polymerase, initiation of transcription. Remember about sigma factor. Then template DNA strand is also there. Growing RNA molecule, template DNA strand, RNA polymerase released. Then termination of transcription, CG-rich stem loop, RNA transcript released, Rho termination protein, Rho protein moves along RNA. Rho protein. Next is about termination. In higher eukaryotes, TTF1 binds and bends the termination site at the 3' end of the transcribed region. This will force pol 1 to pause TTF1 with the help of transcript release factor PTRF and T-rich region will induce pol 1 into terminating transcription and dissociating from the DNA and the new transcript. Evidence suggests that termination might be rate limiting in cases of high rRNA production. TTF1 and PTRF will then indirectly stimulate the reinitiation of transcription by pol 1 at the same rDNA gene. In organisms such as budding yeast, the process seems to be much more complicated. So initiation and then termination. Next is about rRNA synthesis and processing. The genes coding for rRNA except 5S RNAs are located in the nucleolar part of the nucleus. The rRNA genes are highly repetitious and the mammalian cells contain 100 to 2000 copies of the rRNA genes per cell. The genes are organized in transcription units separated by known transcribed spacers. Each transcription unit contains sequence coding for 18S, 5.8S, and 28S rRNA. The transcription units are transcribed by RNA polymerase 1 into giant RNA molecules, primary transcripts that in addition to the sequence corresponding to 18S, 5.8S, and 28S rRNA contain external and internal transcribed space sequences. The rate of nucleolar transcription is very high and many polymerase operates on the same transcription unit. Transcriptionally active DNA therefore has a Christmas tree-like appearance. on electron microscopic pictures. Transcriptionally active DNA has a Christmas tree-like appearance, like this, shown in this figure. So direction of transcription, nucleolar DNA is there, RNA transcription unit, 45S pre-rRNAs, ETS portions, ITs, rRNAs, 38-2S pre-rRNA, and TS non-transcribed spacer, ETS external transcribed spacer, non-transcribed spacer, external transcribed spacer, then internal transcribed spacer. The primary transcript is processed into the mature 18S, 5.8S and 28S rRNAs. The processing involves exo and endonucleolytic cleavage guided by SNO RNAs, small nucleolar RNAs in complex with proteins. The mature rRNAs contain modified nucleotides which are added after transcription by a SNO RNA dependent mechanism. 5S ribosomal RNA is transcribed by RNA polymerase III in the nucleoplasm. Each eukaryotic cell contains a high number of copies of the 5S coding gene, up to 20,000 copies per cell. 5S rRNAs contains overlapping binding sites for two different proteins, ribosomal protein L5 and transcription factor TF3A. The mutual exclusive binding of these two proteins of 5S rRNAs is important for coordinating the expression of 5S rRNA to the production of other rRNAs. Then RNA polymerase 1 completed, RNA polymerase 2. RNA polymerase 2, RNAP2 and POL2 is an enzyme found in eukaryotic cells. It catalyzes the transcription of DNA to synthesize precursors of mRNA and most snRNAs, siRNAs and miRNAs and microRNAs. A 550 kilodalton complex of 12 subunits, RNAP2 is the most studied type of RNA polymerase. A wide range of transcription factors are required for it to bind to upstream gene promoters and begin transcription. Many POL2 transcripts exist transiently as single-strand precursor RNAs, pre-RNAs that are further processed to generate mature RNAs. For example, precursor mRNAs, pre-mRNAs is extensively processed before existing into the cytoplasm to the nuclear pore for protein translation. Then promoter RNA polymerase 2. Most eukaryotes use Tata box. It's a little further away from the initiation start site. In eukaryotes, the promoters are a little more complex. These elements functional analogous to the 10 and 35 in prokaryotes. They orient polymerase and bind proteins. TF3B recognition element, Tata box, initiator, downstream core promoter element. Then initiation. To begin transcription, eukaryotic RNA polymerase 2 requires the general transcription factors. These transcription factors are called TF2A, TF2B and so on. The promoter contains a DNA sequence called the Tata box, which is located 25 nucleotides away from the site where transcription is initiated. The Tata box is recognized and bound by transcription factor TF2D, which then enables the adjacent binding of TF2B. For simplicity, the DNA distortion produced by the binding of TF2B is not shown. The rest of the general transcription factors as well as the RNA polymerase itself assemble at the promoter. So TF2H uses ATP to pry apart the double helix at the transcription start point, allowing transcription to begin. TF2H also phosphorylates RNA polymerase 2, releasing it from the general factors so it can begin the elongation phase of transcription. As shown, the site of phosphorylation is a long polypeptide tail that extends from the polymerase molecule. So core promoter TF2D binds to Tata box in DNA. Then TF2D, TF2A and TF2B form complex with TF2D. TF2A and TF2B resulting complex is bound by RNA polymerase attached to TF2F. TF2F RNA polymerase 2. Then pre-initiation complex is completed by addition of TF2E and TF2H. So pre-initiation complex RNA polymerase undergoes phosphorylation. RNA polymerase 2 is there, transcription begins. Once again, core promoter is there, data box DNA, start point. TF2D binds to data box in DNA. Then TF2D is there, TF2A and TF2B form complex with TF2D. So TF2A and TF2B is there, resulting complex is bound by RNA polymerase attached to TF1IF, TF2F RNA polymerase 2, TF2F, then TF2H pre-initiation complex. Processing of mRNA. All the primary transcripts produced in the nucleus must undergo processing steps to produce functional RNA molecules for export to the cytosol. We shall confine ourselves to a few of the steps as they occur in the processing of pre-mRNA to mRNA. The step, synthesis of the cap. This is stretch of 3 modified nucleotides attached to the 5' end of the pre-mRNA. Synthesis of the poly A tail. This is a stretch of adenine nucleotides attached to the 3' end of the pre-mRNA. Step-by-step removal of introns present in the pre-mRNA and splicing of the remaining exons. This step is required because most eukaryotic genes are split. So DNA, the gene, transcription, intron and exons are there. Pre-mRNA, capping and polyadenylation. Pre-mRNA, excision of introns and splicing of exons. To cytosol 4 is translation by ribosome. Then capping in detail. mRNA capping enzymes, triphosphatase, guanylyl transferase, guanylyl 7-methyl transferase. Then splicing, 5' splice site, exon, branch point, polypyrimidine tract, 3' splice site, UT and UTAF. Then transesterification, lariat intermediate, transesterification to lariat intron, etc. Then cleavage and polyadenylation. Then A to I editing, adenosine, myosin, inosine, A to I editing. Then 5' cap addition. A 5' cap, also termed as RNA cap, an RNA 7-methyl guanosine cap, or an RNA 3,7G cap, M7G cap. Then RNA 7-methyl guanosine cap, or RNA M7G cap is a modified guanine nucleotide that has been added to the front or 5' end of a eukaryotic messenger RNA shortly after the start of transcription. The 5' cap consists of a terminal 6-methyl guanosine residue that is linked through a 5' triphosphate bound to the first transcribed nucleotide. Its presence is critical for recognition by the ribosome and protection from RNAs. So shortly after the start of transcription, the 5' end of the mRNA being synthesized is bound by a cap synthesizing complex associated with the RNA polymerase. This enzymatic complex catalyzes the chemical reactions that are required for mRNA capping. Synthesis proceeds as a multi-step biochemical reaction. Then splicing. Splicing is the process by which pre-mRNA is modified to remove certain stretches of non-coding sequences called introns. The stretches that remain include protein coding sequences and are called exons. Sometimes pre-mRNA messages may be spliced in several different ways, allowing a single gene to encode multiple proteins. This process is called alternative splicing. Splicing is usually performed by an RNA protein complex called the spliceosome, but some RNA molecules are also capable of catalyzing their own splicing. Then editing. Polyadenylation. Polyadenylation is the covalent linkage of polyadenylyl moiety. Polyadenylyl moiety to a messenger RNA molecule in eukaryotic organisms, with the exception of histones. All messenger RNA molecules are polyadenylated at the 3' end. The poly A tail and the protein bound to it aid in protecting mRNAs from degradation by exonucleases. Polyadenylation is also important for transcription termination, export of the mRNA from the nucleus, and translation. mRNA can also be polyadenylated in prokaryotic organisms, where poly A tails act to facilitate rather than impede exonucleotide degradation. Polyadenylation occurs during and immediately after transcription of DNA into RNA. After transcription has been terminated, the mRNA chain is cleaved through the action of an endonuclease complex associated with RNA polymerase. After the mRNA has been cleaved, around 250 adenosine residues are added to the free 3' end at the cleavage site. This reaction is catalyzed by polyadenylate polymerase. Just as in alternative splicing, there can be more than one polyadenylation variant of an mRNA. Polyadenylation site mutations also occur. The primary RNA transcript of a gene is cleaved at the poly A addition site, and 100 to 200 A's are added to the 3' end of the RNA. If the site is altered, an abnormally long and unstable mRNA results. Several beta globin mutations alter this site. Moderate anemia was result. Once again, I will be reading. Polyadenylation occurs during and immediately after transcription of DNA into RNA. After transcription has been terminated, the mRNA chain is cleaved through the action of an endonuclease complex associated with RNA polymerase. After the mRNA has been cleaved, around 250 adenosine residues are added to the free 3' end at the cleavage site. This reaction is catalyzed by polyadenylate polymerase. Just as an alternative splicing, there can be more than one polyadenylation variant of an mRNA. Polyadenylation site mutations also occur. The primary RNA transcript of a gene is cleaved at the poly A addition site, and 100 to 200 A's are added to the 3' end of RNA. If this site is altered, an abnormally long and unstable mRNA results. Several beta globin mutations alter this site. Then RNA polymerase 3. RNA polymerase 3 or pol3 transcribes small non-coding RNAs including tRNAs, 5S rRNAs, U6 snRNAs, srpRNAs, and other short stable RNAs such as ribonuclease pRNA. Then structure of eukaryotic RNA polymerase 1, 2 and 3 contain 14, 12 and 17 subunits respectively. 14, 12 and 17 subunits. 14, 12 and 17. All three eukaryotic polymers have 5 core subunits that exhibit homology with the B' alpha 1, alpha 2 and omega subunits of E. coli RNA polymerase. An identical omega-like subunit, RBP6, is used by all three eukaryotic polymerase, and while the same gamma-like subunits are used by pol 2 and 3. So the three eukaryotic polymers share 4 other common subunits among themselves. The remaining subunits are unique to each RNA polymerase. The additional subunits found in pol 1 and pol 3 relative to pol 2 are homologous to pol 2 transcription factors. Crystal structures of RNA polymerase 1 and 2 provide an opportunity to understand the interaction among the subunits and the molecular mechanism of eukaryotic transcription in atomic detail. tRNA gene, A box, B box. Then promoter for RNA polymerase 3. RNA polymerase 3 transcribes small molecular weight RNAs such as tRNAs, 5S RNAs, and 7S RNAs, 7S LRNAs, U6SN some mcRNAs, and it is also transcribed some AGV, EBV, and many eukaryotic viral genes. The 5S rRNAs and tRNAs genes have promoters within the coding region of the gene. The promoter region 47S and U6HN RNAs more or less look like RNA polymerase 2 promoters with little difference. Though the size of the gene is small, ranges from 160 to 400 pp, their promoters are well defined for transcriptional initiation from their respective start sites in the promoters. Then initiation. The construction of polymerase complex on the promoter pole 3 is unusual compared to pole 2, requiring no control sequence upstream of the gene, instead normally relying on internal control sequences within the transcribed section of a gene, although upstream sequences are occasionally seen. UCSN RNA genes as an upstream data box as seen in pole 2 promoters. Then class 1 typical stage in 5S rRNAs, also termed class 1 gene initiation. TF3A transcription factor for polymerase 3A binds to the intragenic lying within the transcribed DNA sequence, 5S rRNA control sequence, and C block, also termed box C. TF3A serves as a platform that replaces the A and B blocks for positioning TF3C in an orientation with respect to the start site of transcription that is equivalent to what is observed for tRNA genes. Once TF3SE is bound to the TF3A DNA complex, the assembly of TF3B. TF3A serves as a platform that replaces the A and B blocks for positioning TF3C in an orientation with respect to the start site of transcription that is equivalent to what is observed for tRNA genes. Once TF3C is bound to the TF3A DNA complex, the assembly of TF3B proceeds as described for tRNA transcription. Class 2 typical stages in a tRNA, also termed class 2 gene initiation. TF3C transcription factor for polymerase 3C binds to two intragenic lying within the transcribed DNA sequence control sequences, and A and B blocks, also termed box A and box B. TF3C acts as an assembly factor that positions TF2B to bind to DNA at a site centered approximately 26 base pairs upstream of the start site of transcription. TF2B is the transcription factor, and TF2C acts as an assembly factor that positions like data binding protein, the pole 2 transcription factor, TF2B related protein, BRF1 for BRF2 for transcription of a subset of pole 3 transcribed genes in vertebrates, and BDP1. TF3B is the transcription factor that assembles pole 3 at the start site of transcription. Once TF3B is bound to DNA, TF3C is no longer required. TF3B also plays an essential role in promoter opening. So TF3B remains bound to DNA following initiation of transcription by pole 3, unlike bacterial sigma factors and most of the basal transcription factors for pole 2 transcription. This leads to a high rate of transcription reinitiation of pole 3 transcribed genes. So tRNA gene, A box and B box, TF3C, TF3B, TBP, then BRF. Some other processes are also taking place. And then class 3 Typical stages in a U6 snRNA, also termed class 3 gene initiation, documented in vertebrates only. SNAPs, snRNAs, activating protein complex, also termed PBP and PTF, binds to the PSE, proximal sequence element, centered approximately 55 base pairs upstream of the start site of transcription. This assembly is greatly stimulated by the pole 2 transcription factor OCT1 and staph that binds to an enhancer like DSF, distal sequence element, at least 200 base pairs upstream of the start site of transcription. These factors and promoter elements are shared between pole 2 and pole 3 transcription of snRNA genes. So 5S rRNA gene, C box, TF3A, then TF3C, TF3A, then 5S rRNA gene, distal sequence element, at least 200 base pairs upstream site of transcription. This factor and promoter elements are shared between pole 2 and pole 3 transcription of snRNA genes. Then 5S rRNA gene, C box, TF3A, TF3C and TF3A. Some processes are taking place. Then SNAPC acts to assemble TF3B at a Tata box center 26 base pairs upstream of the start site of transcription. It is the presence of a Tata box that specifies that the snRNA gene is transcribed by pole 3 rather than pole 2. The TB3B for U6 snRNA transcription contains a smaller BRF1 paralog, BRF2. TF3B is the transcription factor that assembles pole 3 at the start site of transcription. Sequence conservation predicts that TF3B containing BRF2 also plays a role in promoter opening. Each of the internal sequence represents certain tRNA domains, such as a block representing D-arm and B block representing TCG loop, respectively. At the time of transcription initiation, a transcriptional factor, TFC, made up of 6 subunits, recognizes the sequence boxes and binds to them and positions the proteins in such a way one end of the protein is found at the start site. Then this protein guides the TFB, which is made up of several subunits, to be positioned at start site. Then the DNA RNA pole 3 recognize these proteins and bind to them and binds tightly and initiate transcription at the predefined site. Here, the role of promoter is to provide recognition sequence modules for specific proteins to assemble in such a way the polymerase is primarily positioned to initiate transcription exactly as a predefined nucleotide, which is called the start site. So, if sequence motifs are not present, proteins fail to bind and RNA pole fails to associate with accessory proteins and initiate transcription at specific site. In these promoters, there is sequence just Tata box for the binding of TBP, which acts as a position factor. This is what the promoter is and what it is meant for. This is why promoter is required. So 5S RNA genes, ribosomal RNAs in eukaryotes consist of 28S, 18S and 5.8S and 5S rRNAs. The 28S and 18S and 5.8S rRNAs are synthesized as one block from nucleolar organization region of DNA, and the precursor 5S, 45S, larger than the final RNAs, is processed into 28S, 18S and 5.8S RNA, but no 5S RNA segment. Gene for 5S RNA are located elsewhere in the chromosome. Many times they are found just behind telomeres. The number of 5S RNA genes in a haploid genome can vary from 200 to more than 1200, and all of them are randomly repeated in the cluster, and each of them are separated by non-transcribing spacer. So during transcriptional initiation, TF3A first recognizes the C box and binds, then TF3B containing TBP binds to the promoter using TF3A and it positions at start site. Then the RNA pol 3 complex assembles at the start region and initiates transcription at the predefined site. Again, the role of internal promoters is to position the transcriptional factors and ultimately the RNA pool so as to initiate at specified site. 5S RNA expression differs in oocyte and somatic tissues. Transcription factor TF3A 40 KD protein is produced in oocyte specific manner. This protein binding to internal site of the 5S gene activates the gene expression by facilitating the assembly of TF3C and B and finally RNA polymerase 3. At a late stage of oogenesis, enormous quantities of 5S RNAs are produced and a TF3A binds to 5S RNAs. Thus, all TF3As get consumed and none of the factors are available for the activation of oocyte-specific 5S RNA gene. Then termination. Polymerase 3 terminates transcription at small poly-T stretch 5 to 6. In eukaryotes, a hairpin loop is not required as it is in prokaryotes. Processing, tRNA synthesis and processing. So, tRNA is transcribed by RNA polymerase 3. You know that. The transcription product, the pre-tRNA, contains additional RNA sequences at both the 5' and 3' ends. These additional sequences are removed from the transcript during processing. The additional nucleotides at the 5' end are removed by an unusual RNA containing enzyme called ribonuclease B, RNase B. Some tRNA precursors contain an intron located in the anticodon arm. These introns are spliced out during processing of the tRNA. All mature tRNAs contain the trinucleotide CCA at their 3' end. These 3 bases are not coded for the tRNA gene. Instead, these nucleotides are added during processing of the pre-tRNA transcript. The enzyme responsible for the addition of the CCA end is tRNA nucleotidal transferase and the reaction proceeds according to the following scheme. tRNA plus CTP is tRNA C plus PPI, pyrophosphate. tRNA-C plus CTP gives tRNA-CC to PPI. Then tRNA-CC plus ATP, tRNA-CCA plus PPI. Mature tRNAs can contain up to 10% bases other than the usual adenine, guanine, cytidine and uracil. These base modifications are introduced into the tRNA at the final processing step. The biological function of most of the modified bases is uncertain and the translation process seems normal in mutants lacking the enzymes responsible for modifying the bases.